Hello everybody, welcome back to section 2 of the first section of MMP. Today's lecture topics are glycolysis, beta oxidation, and glucose regulation in the body. So when talking about glycolysis, there's two phases. There's the preparative phase, which requires the input of 2 ATP, and the ATP generating phase, which outputs 4 ATP. Giving a net of 2 ATP from glycolysis. Now, glycolysis begins with glucose, which is converted through a series of reactions until you get fructose 1,6-bisphosphate. Now, this is reacted further through what is called an aldol cleavage, and it will become two molecules. It will become glyceraldehyde 3-phosphate and 1,3-bisphosphoglycerate. Now, you can see here that this reaction actually essentially occurs twice. Just glyceraldehyde 3-phosphate needs to react once into 1,3-bisphosphoglycerate, and then it'll continue with the reaction to 3-phosphoglycerate. It'll go up here, that'll go down here. I know it's all over the place. It'll come over here until finally ending with pyruvate. Things to notice, this step right here generates an NADH. And this is really useful for some processes later on that we'll talk about, as well as the direct ATP generation. And if you remember me mentioning, this aldol cleavage essentially produces two molecules. That's why it's 4 ATP, even though these two right here are the only steps with ATP visible. So it's basically like 2 times 2. So this right here is a completely blank diagram of glycolysis. From both the preparative phase and the ATP generating phase. I'll provide a link for that in the description of the video. Something I want to note is these boxes over here are like your enzymes, these are your substrates, this is where it branches off, and then these are any reaction that produces a product. Now this can range anywhere from something like NADH to like ATP down here. And I think it's just good to learn what those are. This isn't as stringent as the TCA cycle was on remembering enzymes, but you probably will get like 1 or 2 questions about it randomly. I would look at some of the bigger steps as far as that goes, but some reactions, especially like in this region, you probably won't be asked. I'd be pretty surprised. So with that NADH that we generated in glycolysis, there's several things you can do with it. It's a good energy carrier in the cell. As you may remember from last time, it can be used in the electron transport chain for generation of ATP. Something else it can be used for is shuttling malate and oxaloacetate across the mitochondrial membrane. So if you ever need either of these things either inside the mitochondria or outside the mitochondria, you could use NADH in order to accomplish that. Something else you might also notice is we have alpha-ketoglutarate as well as glutamate here. This is important because glutamate is actually a nitrogen-containing compound. And that's important because we can actually use steps like this for amino acid synthesis a little bit later. But that won't be until around the end of the course that we talk about that. As you may or may not remember from last time, the electron transport chain requires oxygen in order to work. There is a way we can get some additional ATP when there's no oxygen available, and that can be by turning pyruvate into lactate. This will generate 2 more ATP. However, compared to the electron transport chain 30 or so, this is not very efficient. This is essentially what happens when you're suffocating. Also, you may recognize lactate as being a precursor of lactic acid. This is essentially why your muscles get sore when you're working out. They're not getting enough oxygen. So this reaction is actually occurring, and that's why it makes it sore, the acidity that's building up in your muscles. Now, glycolysis itself is actually regulated by a few mechanisms. Most of them are by the substrates, as well as the energy produced, although there are a few niche ones. First thing I'm going to point out is this goes back to our first lecture, the pyruvate dehydrogenase complex. This is essentially just showing if the substrate moves forward or not. I already talked about this a little bit. Calcium and lack of energy make it go forward. Energy and high products make it go backwards or make it occur less at least. Something else is ATP right here. ATP right here both slow down this process because we don't necessarily need to initiate glycolysis if we already have the energy. As you can see here, I actually added one. This was a question on either a quiz or an exam we had last year. Phosphofructokinase 1 is an enzyme that contains magnesium. So magnesium actually is also a regulator of this, as well as AMP making it go forward, and fructose 2,6-bisphosphate making it go forwards as well. As well as that, we also have fructose 1,6-bisphosphate. Again, we have a product from earlier in the reaction making the whole reaction go forward. So there's that. And hexokinase itself, which if you remember is the molecule that regulates this step, if there's a high prevalence of it, it will downregulate this whole process. So a little bit complicated, but I would say the main takeaways are if there's energy, it's not going to happen as much, and if there's a lack of energy, it will probably go forward. So now let's transition into beta oxidation and fat metabolism. So something we can do with that ATP we just generated from glycolysis, among other things, is we can actually make a fatty acyl-CoA. And what we do is we take a phosphate off of ATP, add it on to a fatty acid, and we're left with this fatty acyl-CoA. Through fatty acyl-CoA synthase is the enzyme there, and you will need to know that one. So with that, we can move this into the membrane of the mitochondria. Similarly to TCA cycle, beta oxidation takes place in the mitochondria. This enzyme right here, CPT1, will actually then split up the fatty acyl-CoA we just made into CoA and fatty acyl-carnitine. Because while fatty acyl-CoA could diffuse across the first membrane, fatty acyl-carnitine needs a transporter to be transported across this next membrane. And then with CPT2, we actually remake our fatty acyl-CoA. So that entire step was just to get this across two membranes. We just did those reactions to end with what we started with. But then once that's actually in the matrix of the mitochondria, it can work there. So now that our fatty acyl-CoA is in the cell, and I may not have explained this as thoroughly on the previous slide, fatty acyl-CoA is essentially any fat. You can see it can be any length, just as long as it ends with this acyl-CoA group right here. We're trying to cleave it, get it down to its most basic parts, and we're getting energy each time we do that. And we do that through essentially three enzymes. We begin with acyl-CoA dehydrogenase, which will produce us FAD2H. That's worth about 1.5 ATP each. And then once that's happened, essentially what we've done is we've added a double bond in here. The actual chemistry of this isn't incredibly important. The only thing I would really remember, this will definitely be a question on either a quiz or an exam, is that 2 carbons are cleaved for each round of beta oxidation. So I would definitely remember that. I'd say that's the most important part of all of this, as well as some of these enzymes are more important. I would say if Dr. Robbins is going to ask you about them, it would probably be one of these two. As these two aren't talked about quite as much. Could possibly be number 4 as well, beta-thiokinases, just for it's the one that actually cleaves off this acetyl-CoA, which essentially is, this can go into like TCA cycle and be a substrate for that. So that's how you're actually making energy for this, besides the FAD2H. But just going through this process, you can see we make some NADH as well as we have to input a water there. So that's really what I'd remember. These enzymes aren't, again, incredibly important. Just remember 2 carbons, and you're generating substrates, 1 for the TCA cycle, and 2 energy carriers for things like the electron transport chain. This works really well with straight-chain fatty acids. If there's something like a double bond, though, it has to work a little bit differently. It's still the process of removing 2 carbons. It's actually even a little bit simpler, as this took 4 steps over here. This one only took 3. So essentially, once you encounter a double bond at this 2 position here, assuming this carbon's the 1 position, you have to insert a little NADPH. So you're removing some energy to do that, but then you're combining a double bond, or at least moving this one over here, to make that happen, and then this carbon can cleave right here, and then eventually you'll get back to your start. It'll keep doing this whenever it encounters a double bond, um, but essentially, if there's no double bond, it'll just proceed as normal. So how is beta oxidation regulated? So similar to glycolysis, if ATP to ADP ratio is high, as well as if you have these energy carrying molecules and those are high, that's going to downregulate the system. Again, kind of makes sense. If there's high energy, you don't really need to be metabolizing things because you don't need the energy at the time. Also, malonyl-CoA is also regulating the system downwards in the sense that if you have a lot of substrate present or too much, then it wouldn't really make sense to burn it all at once. Fat is meant to be saved. So again, the system tries to regulate itself and not wasting too much of its resources. You might also notice insulin here. This actually upregulates it. Insulin upregulates the um, movement of things like glucose, amino acids into your cell. So if insulin is moving various biomolecules into your cells, it makes sense that you can begin to burn them for energy because insulin is present when your energy levels are low. Um, I also want to note that the brain is a primary user of beta oxidation. Uh, a lot of things with acetyl-CoA occur there. I can use them for energy by generation of things called ketone bodies. This isn't something you're going to be tested on too heavily, although I would say if there's any question about where ketone bodies are metabolized, definitely remember that's in your brain primarily, and that's because your brain always needs an energy source. So that's kind of the reasoning for that. Okay, so now let's talk a bit about signaling hormones of sugar. On the last slide, we talked a bit about insulin. Where does insulin come from? Well, it comes from the pancreas in the section called the pancreatic islets. There's two types of cells, at least that regulate hormones in the pancreas. There's the beta cells, which secrete insulin, and the alpha cells, which secrete glucagon. Now, insulin and glucagon are essentially opposites. Insulin uptakes biomolecules into the cells, whereas glucagon releases. And I say biomolecules because things like triglycerides, amino acids, as well as glucose are all regulated by these and not just glucose. So kind of how does the body respond to glucose levels? So right here we can see a fasting glucose level. This is a lot lower, but then you have a meal. You eat this cake, this slice of cake, which obviously has a lot of sugar, a lot of glucose. So what happens is your body detects this, and you can see that the insulin curves and the glucose curves are very similar. This is like an example of negative feedback because as glucose goes up, there's a little bit of a lag phase where insulin will also go up to get everything in the cell, and then as glucose in the blood goes down, so will insulin. Glucagon's the opposite way. Glucagon was actually working overtime even before eating the cake because there wasn't a lot of sugar in your blood, meaning that glucagon was essentially signaling your cells to release more sugar from your cells in order to move it to areas that were more necessary. So moving it out of things like adipocytes and your liver to areas like muscles, the brain, just places where energy are always needed, tapping into your energy stores basically. So let's go a little bit more in depth into insulin, glucagon, and just how they work on different systems of the body. So with insulin, like I said, amino acids and glucose are brought up into cells, such as the muscle and the liver. And in the case of glucose, it'll go into adipocytes versus amino acids, it'll go into muscles. This makes sense if you think high protein diets and muscles. The muscles obviously need a lot more proteins to function, making things like the actin-myosin complex and make it actually move, whereas something like an adipocyte is just a fat storage cell. So you don't really need the amino acids there, but you can definitely put glucose there. Then in the liver, there's also a need for proteins, so that's why amino acids go up into there. And your liver does a lot of like post-processing of molecules as well. Once you're in the liver, insulin promotes the creation of glycogen, a storage molecule. Similarly, the glucose from that goes into the adipocytes will promote triglycerides. That's like a fat storage cell. Um, and then if there's too much glucose in the liver, it'll actually convert it into fatty acids, which can also go into your adipocytes. So essentially your body wants to maintain like a balance of biomolecules. If you have way more glucose than something else, and that something else is needed, you can definitely convert them in between just for whatever you need. And then in the muscle, glucose can go into glycogen. So glucagon's a bit different. As you can see, insulin pretty much affected everything. Glucagon is not quite the same. So when glucagon's released, amino acids will still move up into uh, places like the liver, but they actually may be converted to something like glucose in the liver, which can then be released. So I might have said on the last slide, a bit of a misnomer. I said that glucagon releases amino acids directly from cells. Not quite the case, but they are converted to glucose, which are then released. So in a way, they are still being used up in the body. They're just being mobilized in a different way. And here, storage molecules like glycogen aren't being produced, which kind of makes sense. And actually, glycogen is being promoted to be broken down into glucose because we need it in the blood. Same with fatty acids, we're not producing them. Our triglyceride storage is, oops. Our triglyceride storage is turned more into fatty acids, go back to the blood. That makes sense. I think the biggest one that you should remember when it comes to glucagon is glucagon does not affect skeletal muscles, which makes sense when you think about it. We always need our energy molecules to be in the muscles, so we're never going to essentially mobilize them from the muscle because that's where they need to go anyways. So while insulin can affect everything, it will never affect skeletal muscle, and I can almost guarantee you that'll be a question in some capacity. So now let's talk about how essentially glucose will affect ATP signaling in a cell, how it is signaled. So glucose will enter a cell through various measures and will do everything we've been talking about before: glycolysis, TCA cycle, oxidative phosphorylation. This increases ATP. Obviously, the byproducts of that is a lot of ATP. What happens next goes into a little bit of cellular signaling. It downregulates potassium leaving the cell. In anatomy, you may remember cell depolarization. Essentially what this is doing, we're building up RK plus in the cell till eventually the cell does depolarize and calcium rushes into the cell. Now what calcium can do is it'll essentially act on vesicles inside the cell in where insulin may be stored. So these vesicles will then fuse with the membrane of cells and release the insulin. So essentially, it's not so much glucose that insulin is monitoring, but rather it begins with the energy in our cells and then the depolarization of calcium into the cells. So just a bit of a note there. And then how do these actually, these hormones regulate cells themselves once they are released? Well, insulin is actually something we know a little bit less about. We have a general idea that glucose goes into cells, clearly, because we need it to work there. And this can be through the opening of transporters. And we believe that insulin binds to a receptor and essentially upregulates things like production of these transporters, synthesis into pyruvate and fatty acids. Again, it's a little less clear, just production of storage molecules. But we do have a greater idea of how glucagon works. And this is something that you will more likely be tested in depth on. So a glucagon molecule binds to this protein called GPCR. This is a G-coupled protein receptor, or G-protein coupled receptor. Essentially what these do is there's a step of cascades. You can see this protein is here at first, but then it will snap off, as well as these two. And then this is something you almost certainly get a question on. This protein right here is called adenylyl cyclase. And what this does is this takes ATP, and this will make this into CAMP, which is essentially just removing two phosphates. But what this does is this allows this to be essentially a catalyst for a lot of other proteins. So this CAMP will activate pKa, which will activate PPK. These enzymes aren't important. You might get asked about PK and PPK, but beyond that, the cascade is a little bit irrelevant. But essentially, we get to this process here, which breaks down glycogen into glucose 1-phosphate. So it's essentially through this G protein coupled receptor going through all these steps until we're making ATP, which can work as kind of a signaling molecule, and that will eventually break down glycogen into glucose, which can escape the cell. Something else to note is that epinephrine or adrenaline and norepinephrine work the exact same as this. You might get a question on that as glucagon. They work on beta receptors, but it also uses adenylyl cyclase. This is a very common signaling mechanism in cells. It just does different things whenever it's activated, essentially. Or CAMP can do different things, but in this case, it will target pKa and then go through this process to break down glycogen and macromolecules. So with that, I think that's everything for this unit. Good luck on your quiz or exam, and I will see you in the next one.